Hi, my name is Brian Kim, Professor and Vice Chair of Research in Dermatology, Director of the Mark Lovell Center for Neuroinflammation and Sensation at Icahn School of Medicine at Mount Sinai. In Vignette 3, I'll be talking about type 2 inflammation, one tissue, many diseases. In previous videos, we reviewed the diverse landscape of type 2 immunity and the unique attributes of the cells and signaling molecules that are involved in type 2 inflammation. As we think about how type 2 inflammation leads to specific disease states, we must also consider how the inflammatory cells and their effector molecules are interacting with various cells within tissues. As the type 2 immune response evolved to counter tissue-burrowing helminths, type 2 inflammation affects a host of physiologies, including expulsive, mechanical reflexes, the epithelial barrier itself, and tissue fibrosis programs to wall off invasion. In this video, we will explore how various cell types in the tissue are affected by type 2 inflammation, specifically in the skin. The skin is a very complex organ with multiple layers forming a dynamic structure that serves as the body's initial barrier against pathogens, UV light, chemicals, and mechanical injury, in addition to regulating temperature and transepidermal water loss. Under healthy conditions, skin-resident stromal cells, including keratinocytes and fibroblasts, coordinate with a variety of immune cells to ensure tissue immunity and homeostasis. However, when this balance is disrupted, a variety of different pathologic processes can be triggered. First, we'll look at the skin's outer layer, the epidermis, which includes sublayers and is composed predominantly of keratinocytes. Under normal conditions, keratinocytes play a key role in maintaining barrier integrity and providing structural support by forming tight bonds with other keratinocytes in the epidermis. While keratinocytes are relatively quiescent at steady state, they can release large amounts of chemokines and cytokines, including alarmins like TSLP and IL-33, in response to barrier disruption. These cascades can become dysregulated in diseases such as atopic dermatitis, leading to the excessive recruitment and further activation of immune cells in the skin. Additionally, proteases such as kallikreins become rapidly upregulated and hyperactive in the setting of barrier disruption, which can further degrade the barrier and activate other immune cells. Downstream type 2 cytokines further impair the expression of proteins involved in skin barrier function, such as filaggrin and tight junction proteins, leading to increased permeability. Collectively, the sequence highlights how type 2 inflammation-associated events can lead to epidermal barrier dysfunction in conditions like atopic dermatitis. Now, let's move deeper in the skin to look at the dermis, which provides structure and support to protect the skin, assists in thermal regulation, and provides sensation for the skin through nerve fibers distributed throughout both the dermis and epidermis. This fibrous structure is composed of dermal fibroblasts, collagen, elastin, and other extracellular components. Type 2 inflammation has significant effects on dermal fibroblasts, key cells involved in tissue structure, repair, and remodeling. Fibroblasts can be activated by IL-4, IL-13, and IL-31, leading to extracellular matrix deposition, proliferation, and differentiation. Type 2 cytokines, along with transforming growth factor beta, or TGF-beta, can stimulate fibroblasts to produce increased amounts of extracellular matrix proteins such as collagen and fibronectin, leading to fibrotic changes. Clinically significant tissue remodeling markers are mostly observed in mucosal epithelial tissues, as seen in type 2 respiratory and gastrointestinal disorders. Activated fibroblasts can also secrete periostin, which can bind to keratinocytes and induce TSLP expression and can directly stimulate sensory nerve fibers to trigger itch. Thus, by integrating type 2 immune response to fibroblast biology, we can start to appreciate how aberrant dermal responses may promote fibrotic pathologies in prurigo nodularis. In addition to fibroblasts, the dermis is a major reservoir of tissue-resident immune cells, such as mast cells, which reside in close proximity to sensory neurons and blood vessels. Thus, we can now appreciate how a rapid event such as IgE cross-linking on mast cells can activate the formation of a hive and itch simultaneously in the setting of conditions such as chronic spontaneous urticaria, while having very little effect on the overlying epidermis. Further, nerve endings in the skin play an important role as they elicit all of the principal sensations: touch, pain, itch, pressure, and temperature. Various inflammatory mediators released during type 2 inflammation, including IL-4, IL-13, and IL-31, as well as histamine and leukotriene C4 and proteases, can directly stimulate sensory neurons to trigger either itch and/or neural hypersensitivity. Further, we are increasingly appreciating the capacity of sensory neurons to produce pro-inflammatory neuropeptides such as substance P and CGRP to trigger inflammation. In fact, substance P directly stimulates MRGPRX2 on mast cells and induces their activation. To summarize, type 2 inflammation interacts with the cells in the tissue in distinct ways, which may lead to the characteristic signs and symptoms of different type 2 inflammatory diseases. In type 2 dermatologic diseases, this includes interactions with skin cells such as keratinocytes and fibroblasts. Additionally, inflammatory mediators interact with sensory neurons to generate sensations such as itch, a characteristic symptom of type 2 inflammatory diseases, and may also trigger neuroinflammation. The relative contribution of these different processes may likely explain the unique features of diseases that still fall within the family of type 2 inflammation.